Madhukar, if you want to share those slides. So I'll spend 5 minutes. I promise I'll be short on who we are and what we do, and then we'll jump into GOBIOM Biomarker Database. Just as an introduction, Exara is a data company that has evolved over the last 15 years from straight data extraction to basically identifying the data inside of patent research article, scientific websites, company pipelines, to an insight company, whereby we've taken that data and extracted and massaged it, wrangled it to kind of get as much information as possible. And that's when we moved into bioinformatics. And then from bioinformatics, we've been able to extract and help company define which target to go after, which target those targets are actually available for in an indication, what kind of biomarkers are related to that target and to that indication, and then diving into actually helping with lead opt and then all the way into the clinics. How do we get a company or a pharma company or an academic research institute to go from, okay, I have my target, I've done my lead up, I now have this compound, how do I take this, how do I translate it from research into development? And then over the last couple of years, we've now expanded ourselves beyond the bioinformatics into the R&D informatics. And that's really helping the IT space learn more about, what do chemists and scientists and biologists do, and how do you give them access to the best technology to be able to empower them to make faster decisions? And so that's where we're starting to get into cloud migration, going from on-site servers to the cloud, where we move into ELN and LIMS. How do we help you implement the best ELN solutions for your needs? And so we're slowly going after the areas where we find scientists and technology at times can struggle to understand each other, and that's where we try to bridge that gap. So data, insights, and enabling the teams to actually be able to use that insight so that they can make use and go faster with their discovery. And that's who we are. We've been in the business for about 2 decades. We work with top pharmas, we work with biotechs, we work with academics. We have a very large workforce of PhD people that are expert in their fields. And we have offices now across the globe. So we've got offices in India, we've got offices in Europe, so Belgium, Netherlands, England, and also across the US. I'm in Boston, and then some of the team is in the San Francisco Bay Area. That's just kind of a view of who we are. And then Madhukar, I know you keep flicking to the next slide, so I'll go there. Just to give you a sense of how we help through the value chain, or how we go through the drug development process from discovery to translational to clinical, and then finally getting you to that value evidence. So a lot of different offerings, kind of giving you a sense of we do a lot of things. But again, we always go back to those 3 things: high quality data, providing insights that you can actually take your research to the next level, and then making sure you're enabled with your technology to make sure you can make the best of it. And that's really it. Now, today we've got myself, Norman Azulay, a director of product at Exara. Been at Exara for 3 years. My goal is to find ways in which I can enable my users to get more out of our data, as well as providing off-the-shelf offerings. So come in with a question, leave with an answer, and repeat, rinse and repeat. And so we do that today for biomarker. That's what we'll see today, but we'll also do it for small molecule. We have a GoStar database, and then we're trying to now expand into the monoclonal antibody areas, as well as degraders, as those are becoming more and more interesting spaces for discovery. So we've got myself, we've got Madhukar, who's been with Exara for almost 20 years. He's been doing GoBiome demo for a long time and is our internal expert. We've got Aparna, who's our biomarker / diagnostic SME at Exara. She also leads our strategy for product, and she helps me build that vision, find out where we're going next. And so she's got some cool stuff to show you towards the end of this call, if we have time, around where we might be going next with biomarkers. And then Nakita is on the business development side, helping us, making sure we stay in touch with our customers and making sure we advance any business discussion. So with that, over to you, Madhukar.
Thanks, Norman. Hello, everyone. Thanks for joining the seminar. Today, we'll be showcasing our GOBIOM Biomarker platform. I have a couple of slides which will provide you a brief overview on the product, following which I'll be demonstrating our online platform as well. GOBIOM stands for Global Online Biomarker Database. So it is an online platform. We do have an active UI or application when you can interact with the data. And you can also opt for a flat file, which is offered to the clients based on their requirement in different formats as well. So if you look at the GOBIOM database, it is a repository of 3 types of biomarkers. We have the clinical biomarkers, exploratory clinical biomarkers, and the preclinical biomarkers. So if you look at the clinical biomarkers, these are a set of very well evaluated biomarkers which are being studied as part of the clinical trials. So we have categorized these biomarkers as clinical biomarkers. So in the same clinical space, we do have a few biomarkers which are being reported in small clinical populations by researchers and academia. So these biomarkers have preliminary information on their clinical qualification in clinical samples, but as such, there is no evidence of them being studied in any clinical trial. So though they are being studied in clinical samples, they are not still being included in any clinical trials. So we have categorized these biomarkers as exploratory clinical biomarkers. And the third category of the biomarkers are the preclinical biomarkers, as the name suggests. These biomarkers are predominantly studied in preclinical models, and there is no evidence of them being studied in humans so far. So these are categorized as preclinical biomarkers. And if you look at the left-hand side of your screen, we have adopted a proprietary tetrahedron model for the GOBIOM database model, wherein the biomarker, disease, drug, and the drug target, they form 4 vertices of the tetrahedron. And GOBIOM being a relational data model, we try to relate each and every point in the tetrahedron to one another. For instance, for the biomarkers which are indicative of disease progression, prognosis, or diagnosis, we try to link the biomarker to the disease. And wherever there is a mention of an intervention or a drug, we try to link the biomarker to the drug and its target. Coming to the database content, as you can see here, the information is collated from varied data sources. But having said that, majority of the data is coming from the different clinical trial registries and the PubMed literature database as well. Apart from that, we also collate information from FDA, EMA, and the PMDA approved documents. And we also include information on approved diagnostic assays from 510K and the PMA database, which have information on the approved class 3 medical devices. And we also collate information from different scientific conferences across the globe. As on today, we cover around 87 scientific conferences, from wherein we visit their websites and collate the information on the biomarkers from the abstracts and the posters which are published on their websites. Last but not the least, we do collate information from the patents, but having said that, we carefully screen the patents because most of the patents have information that is coming out from a large-scale microarray or GWAS studies, wherein there is a reference to differentially expressed genes, but we carefully screen the patents and only curate the data which have experimental information relating to biomarker in there. So if you look at the highlights of the GOBIOM database, the entire GOBIOM data is manually curated, and we update the data in the database on a monthly basis. And throughout the database, we follow standard nomenclature. For instance, for nomenclature pertaining to the diseases or the therapeutic areas, we follow ICD-10. Nomenclatures relating to proteins and genes, we follow NCBI nomenclature. And the nomenclature relating to the drug-related events, we follow MIDRA as well. Not only that, wherever standard ontology is not available, for certain data fields like outcomes. We follow our own Excel proprietary ontologies to ensure that all the data fields in the database are standard throughout. As mentioned earlier, GOBIOM is a relational database which has an intuitive search engine, which I'll be demonstrating to you shortly. Using the online interface, you can download the data into various flexible downloadable options like an Excel, PDF, or an XML document as well. Very shortly, I'll touch base on the process that we follow internally, right from identification of the biomarker from various data sources to integration of the data into the database. So the process starts here with identification of the biomarker in a given disease from varied data sources. When I say data source, it could be a clinical trial, it could be a paper published in PubMed literature database, it could be a patent, it could be a regulatory document, or it could be coming out of a scientific conference as well. So once we have identified a biomarker in a given disease, we classify the biomarker based on the nature. So here we assign the nature to the biomarker based on the parameter which is being measured. For instance, if anything related to the gene, that is the gene expression, the microRNA polymorphism or mutation is being evaluated as a biomarker, we categorize such biomarkers as genomic biomarkers. And if protein itself is being evaluated as a biomarker, it is categorized as a proteomic biomarker. And if a biomarker is measured in blood or urine and measured biochemically, we categorize those biomarkers as biochemical. And if a cellular component is being measured as a biomarker, it is categorized as cellular. And if any anatomical or physiological parameter is being measured by imaging method, we categorize those biomarkers as imaging biomarkers. And if a physiological response of a body is being measured as a biomarker, it is categorized as physiological. And we have also included scoring scale as one of the biomarker classifier. So these are biomarkers which are specific to certain diseases like pain and mental disorders, wherein these biomarkers are used to measure disease severity and the quality of life. Once we have classified the biomarkers based on the nature, we try and identify the utility or the application of the biomarker in a given disease. So to put into simple words, we are looking for the specific inclusion. Why is the biomarker included in a given study? Here we try to identify various utilities relating to the biomarker, like diagnosis. If a biomarker is differentiating a normal population from a disease population, if a biomarker is indicative of any future events, we categorize it as a prognosis biomarker. And if a biomarker is linked to any disease progression, it is categorized as progression of disease biomarker. And if a biomarker is indicative of any drug-related events, it is categorized as a safety toxicity biomarker. And if a biomarker is discriminating drug responders from non-responders, we categorize those biomarkers as response to therapy biomarkers. And if a biomarker is a surrogate to a clinical endpoint, it is categorized as a surrogate biomarker. And if a biomarker is indicative of a drug-related efficacy, pharmacokinetics, or drug resistance, we assign utilities relating to efficacy, pharmacokinetic, and drug resistance as well. So once we have classified the biomarkers based on the nature and identified the utility or application of the biomarker in a given disease, we try and look for any supporting evidence that is available for the given biomarker disease combination in the public literature. So here we try to look for any availability of any experimental assays or methods that are available to measure the biomarker. And if assay is available, we try and look for information like sensitivity, specificity, and the accuracy of assay, that is the analytical qualification of the experimental method. And we also look for the clinical sensitivity of the biomarker for a given disease. And if supporting preclinical data is available, we try and collate that information as well. And if a biomarker is evaluated under different medical interventions, we try and include the information on the drugs, like the drug doses, the duration of the treatment, and the biomarker outcome after the treatment. The recent inclusion to our curation portfolio are the clinical practice guidelines issued by different societies and the drug labels as well. So once we have identified the literature, we add this literature to our curation pipeline, and this literature is assigned to the individual curators, the data scientists who curate the data using our in-house curation tool, which has ontologies embedded into it. So no matter how many data scientists are curating the data, the output is pretty much standard. So following the curation, we have regular rounds of review and QC to ensure that quality data is being fed into the database. And once the data is integrated into the database, the end users can access the data using our online user interface, or they can also request for a customized data feed or a data dump, which is available in different file formats. So you can integrate this dump or the data feed behind your firewalls, and you can integrate your internal data for further analysis as well. A snapshot on the statistics of the GOBIOM database. We have close to 213,000 biomarker indications in our database. Across 18 therapeutic areas, we have curated information on 6,500 odd diseases from 200,000 odd references. And we have information on 12,000 odd compounds and 2,000 assays as well. I'll take a short pause here to check if you have any questions. Okay, so if you don't have any further questions, let me move on to demonstrating our online platform. You can access our online platform at gobiomdb.com. So this is a landing page of the online user interface. So you'll be needing your username and password to log in into the database. You can just go ahead and click on the customer login here. And once you provide your access credentials and click on the submit button, you'll be redirected to the landing page. On the left-hand side of your screen, you'll be able to see the statistics pertaining to the 18 therapeutic areas that we have covered so far. So for each therapeutic area, you'll be able to see the information on the diseases, applications, clinical trials, drugs, and the approved assays. And on the right-hand side of your screen, you'll be able to see the different features of the database. So in order to query the database, we have different search options. You can query the database using the quick search, basic search, advanced search, or if you have a list of IDs that you want to search, or if you want to do a bulk query, you can use the batch query option as well. And you can instantaneously generate reports as well. You can generate a report on a disease, drug, or a biomarker. And on top of that, we do have different analytics where you can compare two different data sets using comparative analysis. You can generate heat maps using Heatmap Analytics. The dashboard analytics feature allows the user to quickly generate dashboards on a disease, drug, or a biomarker. And we also have a dedicated variant analysis platform and a chromosomal view option for the gene variant analysis as well. So in order to query the database, you can use the search option. As mentioned earlier, we have different search options. You can use the quick search option it's a Google-like search option wherein we have a single search box wherein you can search for a biomarker, disease, or a drug. And then we also have a basic search option wherein, as the name suggests, it is a basic level search option wherein you can search by a biomarker name, disease name, or a drug name. At the onset, you can also restrict your search output based on the biomarker clinical status. That is, you can select your search output for clinical, exploratory clinical, or the preclinical biomarkers as well. So by default, all the three options are selected here. And we also have a batch query option wherein you can query the database using the different IDs. For instance, if you were to search by entry symbol or entry IDs, you can just select the parameters here, copy and paste the values here in the search box. Or if you have a list of clinical trial IDs or a PubMed IDs, you can copy and paste the values here. Or you can also upload the data in a text file format using the upload. And click on the submit button. And we also have an advanced search option. As the name suggests, it is an advanced version of the basic search, wherein in addition to the biomarker, disease, and the drug module, you can search by the clinical module, wherein you can search by a clinical trial ID, phase, the trial completion status, or the study population ethnicity. And we have a phenotype module wherein you can search for association of the biomarker with a particular phenotype with statistical significance. And we also have an assay module where you can search by the specimen details, the experimental method, or availability of the assay kit or the companion diagnostic as well. And we also have a preclinical module where you can search by model name or cell line information. And you can also restrict your search output based on the publication type or the year of publication as well. So in order to query the database, all you need to do is just key in a few characters. For instance, if you were to search for a particular therapeutic area, say oncology, you can just type in a few characters and the tool will populate the matching entries for you as a dropdown. You can just select the entry from the dropdown, or you can also input the wildcard character star, or the tool will display all the entries associated with that particular field. You can just select the entry from the dropdown here. So once the selection has been made, you can see that selection here on the top of the table. And once you have selected a parameter from any of the given fields, there is a progressive filtering that is going on in the backend. And due to the progressive filtering, if you now go to the disease name field here and input the wildcard character star, you'll only be able to view the diseases that are specific to the selection that you have made earlier. So here you have selected oncology in the therapeutic area. So since you have selected oncology here, in the drop-down, you'll only be able to view the indications that are specific to the selection that you have made earlier. So this is due to the progressive filtering that is going on in the backend. You can select the parameter from the drop-down here, or you can also start typing the disease of your interest. For instance, if you were to search for ovarian cancer, you can just select ovarian cancer from the drop-down here. And once you have selected the parameter, you'll be able to view the parameters that you have selected here on the top of the table. Similarly, you can apply filters in any of the given modules. For instance, if you were interested in a particular biomarker type. So let's go to the biomarker type here in the biomarker module. And if you are interested in a particular biomarker type, say proteomic and genomic, you can just select genomic and proteomic. Multiple selections are allowed here. I'm selecting genomic and proteomic here. Similarly, you can apply filters based on the application. If you were interested in a particular application type, say diagnosis, safety, or efficacy, you can just select the biomarker type here, application type here. And in the clinical module, you can specify the clinical phase, completion status, or ethnicity. Similarly, you can, if you're looking for biomarkers evaluated against a particular drug, you can search by a drug name or drug target as well. And as mentioned earlier, you can also specify the phenotype or the statistical significance. And if you are looking for biomarkers measured in a particular specimen, you can also specify the specimen here, or if you are looking for biomarkers measured by an experimental method, You can also select the experimental method here. And if you are into preclinical biomarkers, you can select the model or the cell line name. And you can also select the publication type. So if you are looking for biomarkers coming out of clinical trials, you can just select clinical trial and clinical trial result here. And last but not the least, you can also segregate or restrict your search output based on the year of publication as well. So if you look at the selection that I have made here, I've selected the therapeutic area as oncology, the disease as ovarian cancer, and the biomarker types as genomic and proteomic. So you can just go ahead and click on the search icon here on the right-hand side of your screen. So once you click on the search icon, you'll be redirected to the search results, which is a combination of visualization, form tree visualization, and a tabular results view. So if you look at your search results, using the form tree visualization, you can further filter your result set here by just clicking on the parameter that you're interested in. For instance, if you were to apply filters based on the phenotypes, you can just select the phenotype in the form tree visualization here. Or you can also use the applied filter section here to further get to the subset of data. And if you look at the search results here, this is a tabular view of the search results. And you can see here the information on the biomarker name, disease name, biomarker type, biomarker class, and biomarker status, the approval status, the different applications pertaining to the biomarkers, the interventions, number of clinical trials, and the number of references particular to a given biomarker disease combination. So for every biomarker disease combination that we curate in our database, we assign a unique ID to it, which we call as Accelera biomarker ID. And if you were to look at the information relating to that biomarker disease combination, you can click on the hyperlink here, which I'll be showcasing to a later time point. And if you look at the search results here, we have followed a set of criteria to display the search results here. So the biomarkers which are having approval information will be on the top of the table, followed by the biomarkers which have maximum number of citations published on them. So this is the criteria that we are following here to display the search results. So now if you were to look at the information relating to a given biomarker, you can click on the EBM ID here, and you'll be redirected to what we call as a comprehensive biomarker disease report. So here, this is a report which has comprehensive information relating to the given biomarker disease combination. As you can see here, in the reports on the GOBIOM application, we try and segregate the data carefully into different sections. For instance, if you were to look at the data relating to approvals relating to the biomarker, so that you'll be able to find here in the approval section, and the information on the clinical guidelines. Here you'll be able to find the information on the approval authority, the recommendation status, and the brief description on the guidelines, and the direct link to the information from there where this data was collated from. You can just click on the link here to view the reference information.
Madhukar, there is a question in the chat box. Is it possible to enter biomarker set and test list of diseases that the combination of biomarkers are associated with?
Okay, so the question is relating to the combination search. Currently, you cannot enter the combination, but for the panel of biomarkers, we do have annotated that data as a panel of biomarkers in our database. So you can just select the panel of biomarkers from the biomarkers list, and then you can combine it with the disease of your interest. I hope I've addressed your question. Okay, so now coming back to the disease report, the biomarker disease report, you can see here in the diagnostics section, we do carefully include information relating to the diagnostics, like the diagnostic kit name, the kit vendor information, approval status, approval authority, and if it were a companion diagnostic, we do provide information on the associated drug and direct link to the regulatory document. You can access the regulatory document by clicking on the link here. And the application section here provides you the information on the application of CH125 in ovarian cancer. So all the visualizations that you see here on the GOBIOM application are dynamic. So if you were to view information relating to any of the given data points, you can just click on it. So if you were to look for the information relating to this data point, you can just click on it and there will be a pop-up which will provide you the information relating to that data point. And the experimental section here provides you the information on the experimental data. Here you'll be able to view the information relating to the marker alteration, what exactly is happening to the biomarker, and the significance in the p-values, and that link to the reference from where this data was annotated from. The detailed view link here will provide you the information on the details pertaining to that particular data point.
So while you're working on it, Madhukar, the question that you've asked Joanne, I think we still do have the ability of batch entry of biomarkers in GOBIOM. Is that right, Madhukar?
Yes, Aparna. You can actually input that in the batch query option. Yes.
Yeah. So Joanne, if you have a list of biomarkers or genes that you have identified through pathway analysis or some other analysis you've done, you can stick them into GOBIOM, and then GOBIOM will churn out all the diseases that they are linked in the tabular form that Madhukar has been shown.
Thank you. Referring to the phenotypes section here, so here this section provides you the information on the association of a biomarker with a particular phenotype, along with the information on the significance and the p-value. And you can find the data pertaining to the drugs in the drug therapy section, and the information on the marker alteration following the drug treatment. That kind of information is available here in the drug experimental section. So here you'll be able to find information on the biomarker level, the significance, and the p-values as well post the treatment. And the drug phenotype section here provides you the information on the phenotypic changes post the treatment, and along with the information on significance and the p-values as well. And if your biomarker is correlated to other biomarkers, that information is available here in the marker correlation section. And the clinical qualification section here provides you the information on the sensitivity of the biomarker in a given disease. So here you'll be able to find information on sensitivity, specificity, positive predictive value, negative predictive value, and accuracy. And the analytical qualification information pertaining to the experimental method, you'll find that information here in the analytical qualification section. And the information on the clinical population is available in the clinical population section, wherein you'll be able to find information on the sample size, age, gender, information on the ethnicity, stage, and the grade as well. And if preclinical data is available, you'll find that data here in the preclinical section. And you'll be able to find the information relating to the clinical trials here in the clinical trial section. Here you can find information on the clinical trial ID, the phase, study status, the current status, and the direct link to the clinical trial as well. And all the references of the bibliography information is available to view here in the reference section as well. So for every reference that we curate in our database, we assign a unique ID to it, which we call as a reference ID. And you can access the information relating to the reference ID by clicking on the hyperlink here. So all the reports that you see here on the GOBIOM application are searchable and sortable. You can sort the data. And if you are looking to search by a particular keyword, you can also search by the keyword as well using the search box here. Similarly, you can also generate a report on a drug by specifying the drug name here. And you can also generate report on a disease as well. And you can also generate a biomarker report as well. So if you look at the reports in particular, all the structure of all the reports are similar, wherein, as you have seen earlier, we try and segregate information relating to a biomarker disease or a drug into different sections, as you have witnessed in the biomarker disease report. And relating to your earlier query, as specified by Aparna, you can use the batch query option if you were to search for multiple biomarkers or multiple biomarker IDs. If you have a list of entry symbols or entry IDs, you can just copy and paste the values here in the search box, or you can use the upload option here to upload the list of biomarkers in form of a text file. Now that we have also seen the reports, on top of that reports, we also have different analytical options. So from the search results, if you were to save your queries, the tool also allows you to save your queries, and you can retrieve your queries from My Searches here. So if you save your query, for instance, the earlier query relating to ovarian cancer, the biomarker type is genomic and proteomic. So you can see that we also have a save query option. So if you happen to build a complex query, you can save your queries onto the application by using the save query option here. And once you have saved the query, you can retrieve that queries from My Account section here from My Searches. So you can just click on My Searches here. And once you click on that, at a later time point, if you happen to log in into the application, and if you were to go to the search results directly, you can just click on the link here. You can click on open here, and you'll be redirected to the search results. So this will save your time considerably in building the query. So you need not build the query again here. You can directly go to the My Searches and access all the search queries that you have saved before. And from the search results, if you find some biomarkers of your interest, and if you were to download the data, all you need to do is just select the biomarkers of your interest. For instance, if you are interested in a set of biomarkers, all you need to do is just select the biomarkers here. And once the selection has been made, you can view the different options for download here on the right-hand side of your screen. You can download the data in two formats. You can download the data as a flat file, Excel flat file, or you can also download the data as an XML document. So in Excel as well, or in the XML, you have two different options. You can download the summary level information that you happen to see here on the online user interface using the display view option, or if you were to download the data as a report that I have showcased to you earlier, you can use the detailed view option here, wherein the information relating to the biomarker is segregated into different spreadsheets. So the information on the experimental data, the analytic data, the clinical qualification data is segregated across different spreadsheets in a detailed view format here. And you can also download the data into XML format as well.
Madhukar, there's a new question. Is it possible to select specific tissue or blood for biomarker?
Sure. So let me go back to the advanced search here. So let me select the same input here. So I have selected a particular disease name here. And if you are interested in a biomarkers evaluated in a given specimen, say blood, you can just select blood here. Or if you are looking at tumor tissue or tumor fluid, you can also search by a specimen as well. Or you can also search by a given experimental methodology as well. The tool does allow you to search by specimen, methodology, or by the availability of the assay kit or companion diagnostic as well. Okay. So now let me showcase the different
Madhukar, sorry, there's a follow-up.
Yep.
How complete and current is your database? Did you evaluate the quality of studies included in the database?
So Aparna, do you want to take that?
Yeah, the database is updated every month. That's the target. So depending on what we are curating, right? Some of the PubMed articles go on to maybe 2 months delay, but there's a monthly upgrade to the database. The second part of the question was, did you evaluate the quality of studies included in the database? So most of the studies, like Madhukar mentioned, are sourced from clinical trial registries, some with results, some without. The PubMed articles we curate are, so if you mean whether we give any weightage to the journal that the PubMed article is coming from, I suppose not. So every PubMed article which fits into our scope, that is, it carries a biomarker, it is for the disease that we are curating, we curate that, we include that. Yeah.
Thanks, Aparna.
Madhukar, let's break. I think we're at 47 minutes in. Let's open it up for question and see if the audience has any additional question for us. If no other questions, I think what might be of interest is to just show you kind of where we might be going in the next year or so in terms of how do we go further than just identifying biomarkers and how those biomarkers may be of interest and identifying more of a correlation between inference and outcomes for biomarkers. Aparna, do you want to take over?
Okay, so good afternoon, everyone. And like Norman said, it's Friday, so I hope to show you something interesting. What I'm talking about is, and I'll take about 10 minutes of your time to walk you through this, what we are moving ahead with to provide a better takeaway for our clients from our biomarker data. We refer to it as biomarker insights. I hope my screen is visible to everyone. Right, and so I'm talking about biomarker insights. It's a new offering, and you can consider it as a supplement to GOBIOM, which Madhukar has walked you through. What is the new thing that biomarker insight brings is, firstly, we have increased the scope of curation. We are now including more research articles coming out of PubMed. And the focus for these research articles is, of course, the presence of a biomarker, the disease of our choice, and we restrict ourselves to only clinical human sample studies. So all our inferences and outcomes that we associate with the biomarker are coming from studies which have translated into human, either patients or human samples. The other mandate for biomarker insights is to ensure that there is very clear interlinkage between biomarker disease drugs and clinical outcomes. So each and every biomarker has data at least definitely around a clinical outcome linked with the disease. And if there's a drug available, then the drug. And we have tried to portray the data in a much more visual format and with an interactive interface. That's what we are, it's work in progress, and that's what we are attempting to do, so that people are able to probe the interlinks between these 4 core entities very easily, and are able to appreciate the data with visual infographics. So with that, let me show you what we are developing. Okay. Is this okay? So let me sort of show the layout to you here. What you have, this is the biomarker insight page. It's still under development, and what you have on the top left is the count of biomarkers. Currently, what you are seeing on this page is data exclusively for colorectal cancer. So if I use the drop down on diseases, you'll see only colorectal cancer here. So what I know clearly is that I have around 4,000 biomarkers for colorectal cancer. Further, what you see in this sunburst graph is what Madhukar has talked about. Each biomarker is linked with the type and nature. So you have genomic biomarkers, proteomic, cellular, biochemical. You also have microbes, which serve as biomarkers. So we have different kind of biomarkers. And then within genomic, of course, you could be measuring the mRNA, you could be measuring the genetic variant, microRNA, etc. And then you have an application with it, could be used in diagnostic, prognostic. So let's say if you are interested only in looking at risk biomarkers, you'll come straight away and you'll click on show me the susceptibility risk biomarkers, go here and look at what's happening. Okay, then if I go to the dashboard on top, there are 4 different components here. There's disease, biomarker, drug, and outcome. They're color coded, and the same color is sitting in the interactive graphs here. The pink is for biomarker, this yellow one is for the drug, the black one is for the clinical outcome. And in these 2 network graphs, you will see how the biomarker is linked to the outcome and in context with which drug. So if you see them simultaneously, you are sort of able to appreciate that. So these graphs are interactive. If I click on something here, the graph changes, the network changes, and it shows me new connections, which are limited now to genomic because I selected for genomic. Similarly, if I'm looking for only predictive markers, I click here, and the network changes to show me only predictive markers, all limited to colorectal cancer. Note here the number is changing because I have filtered down my biomarkers. So now I reset it to 4,000 biomarkers. I'm looking at everything. I'll just walk you through with some examples so that you can see how you will be able to use this interface, right? So let's say you're working on microbiome, right? So you want to find out which microorganisms are linked to colorectal cancer. Do they increase the risk? What are the sort of clinical outcomes, right? So I've selected only microorganism, biomarker type and nature. I have 28. I zoom into this. And I see there are 28 biomarkers. The black dots are the clinical outcomes that these biomarkers were measuring. So I see the biomarker Fusobacterium nucleatum, that's the microbe, and it's connected to several different outcomes. Let's say I'm interested in figuring out what about progression-free survival, right? This microbe is linked with progression-free survival. I click on it and I find out which drug was it that was involved in that study where this bacteria helped me measure progression-free survival. And I can see that here, it's several arms were there in the study. The pie denotes a different arm, and it was chemotherapy plus some targeted therapies which were involved. Similarly, I can click on, for example, a drug action. Which drug action was it measuring? And I know it was capecitabine plus radiotherapy. So these 2 graphs and all of these work together to tell you in a snapshot what's happening, which drug, which outcome, which biomarker. Let me give you another example. Let's say you have your favorite gene, right, coming out of some study that you're doing, and you have homed in on a particular gene and you want to find out what's happening around that gene. I'll take an example of beta to microglobulin, right? So B2M, I have presence in the form of a variant that's mutants of B2M. So I click on that. I have it as a gene that's beta 2 microglobulin. I also have it in a panel where the researcher had looked at B2M along with JAK1 and JAK2. So I select that also. And then I go in and zoom into the interactive network. And I find that the beta 2M gene here, you can see it's measured both as a gene as well as as a protein in some cases. It's linked with immune cell infiltration, recurrence, clinical response, right? There was clinical response. That's the panel is linked with clinical response. There's the mutant B2M mutations are linked with clinical response. So which drug for clinical response? And I have the answer for that right here. It's these biologics which are associated with that outcome. Similarly, there is something like overall survival. Which drug for overall survival? I know, and I know that there are two drugs here linked with overall survival measurement through BTM variant. There's pembrolizumab, and there's also the chemo with targeted therapies. So that's how this is helping. Another quick example, if you do not have a biomarker in mind, but you have a drug in mind, right? Let's say you're trying to understand why a particular drug is failing, or why, or you want to develop something better than that drug. I'll take an example of cetuximab. So I start typing. There are several studies with the presence of cetuximab in combination, but to save time, I'm just going to go to directly those studies which had only cetuximab. And again, I see a graph here with several outcomes. I can also zoom into it here. And I see that a lot of biomarkers were linked with treatment response. And I know here that the drug in question is cetuximab. I see a lot of them are variants, are mutations in BRAF, KRAS, PIK3CA, etc., etc. And here, this one here is linked, G13D KRAS mutant is linked not just with treatment response, but also resistance. It depends on what drug or what cohort of patients was being looked at. So in a snapshot, you get this view out of the selection. Go back to the report. And the last example that I would show you is coming from outcome, right? Let's say you, as an example, let's say you have, you know that the current unmet need for solid tumors, for example, is that there's poor infiltration of the solid tumors. I'm sorry, most of my examples are coming from oncology. I'm sure people sitting here have worked on a variety of indications, but since I have colorectal here, my examples are picked from that. So let's imagine that there's a situation where you, there's poor immune cell infiltration and you want to find out biomarkers around that. So you go to immune and I select everything with immune, right? Immune cell infiltration, immune evasion, immune response as an outcome. And I get this here where I see immune cell infiltration. There are a lot of biomarkers linked with immune cell infiltration. And let's say I further want to find out how many of these biomarkers which are measuring immune cell infiltration are also impacting overall survival, right? The final outcome that you want to study. So I'll go here and I'll pick up. So I'll just take a minute more. So this, for example, here is an example where the outcome is overall survival. You have a load of biomarkers linked with it. And this is the second outcome, immune cell infiltration. And here you have a subgroup of biomarkers linking both them. So you could have potential interest or a potential new target sitting here, which you want to explore as a combination or, to derive some pathway out of it. So that's it. I think I'll leave you with that. Stop share. And if there are any questions for Madhukar or me, you're most welcome to ask.
Okay. Thank you, everybody. Really appreciate your time. Have a wonderful weekend. Bye.
Thank you. Bye.